Hereby, I open this academic ceremony in which David Boaventura Gomes will defend his academic thesis, Engineering the Bone Marrow Microenvironment, How Biophysical and Biochemical Properties Modulate Stem Cell Behavior. May I invite you to give a presentation of your and a summary of your study and the conclusions of your work.
Thank you so much for the introduction, dear prorector, dear members of the committee, dear family and friends. Today I'm here to present the results that I've been conducting in the past six years of my thesis, which is entitled Engineering the Bone Microenvironment, How Biophysical and Biochemical Properties Modulate Stem Cell Behavior. And to start this journey with you, I would like to introduce you to this character that you all know. This is Super Mario back in the 80s. And as you can see, it's a very pixelized Super Mario with not much definition, and we cannot extract much data about it. Well, Super Mario lived in this 2D world, very caged in, and it could not go backwards. It could only go forward and do basic abilities. Well, thank God this has changed recently. Now Mario is in a 3D world. He can do all the activities that he likes, as you can see by his face, and he can go backwards, and he can go forward, and he can precisely decide his fate. Well, why did I say this? Because this is the same thing that has happened actually back in the 80s, this shift between 2D cultures, where cells are also entrapped, they cannot do much about the space that they have, and they are limited in their actions. Then we move forward, indeed, in the 80s, to cells grown in 3D, where cells have a more natural environment. And of course, you can imagine 3D Mario looks at pixelized 2D Mario and says, well, thank God we changed. And that's the starting point of my thesis, because stem cells, for example, muscle stem cells, hematopoietic stem cells, liver and mesenchymal stem cells, they all have been grown in vitro in 2D cultures, and they lose their ability to be a stem cell, and they become, for example, differentiated, meaning specialized in other tissues. They become senescent, which means that they die, or they proliferate, which are behaviors that they were not supposed to have. So the hypothesis of my thesis was, what happens to a mesenchymal stem cell when it's grown in a 3D culture, when it gets a more natural environment for them? And to start by that, I would like to introduce you what a mesenchymal stem cell, or actually what a stem cell is. And as you can see here by the picture, we have the stem cell in here, and there are two properties that are essential. One, these cells give rise to different tissues, for example, chondrocytes for cartilage or adipocytes for adipose tissue, and this is called multipotency. But the stem cells can be and have these properties throughout all our lifespan, and they do so by a system called self-renewal, where they give rise to another stem cells, and we have homeostasis of our tissue due to that. And this all happens in a place called stem cell niche. Stem cell niche has specific properties, and these properties are regulated by, for example, biophysical properties, or stem cell and ECM, extracellular matrix interactions, or by soluble factors, and even by cell-cell communications. And here we are going to focus on these three. So we decided to create this artificial microenvironment by using a material called alginate. This is a natural material, comes from a natural source, comes from green seaweeds, and we form this sort of hydrogels as droplets, as you can see here. But this is basically the same as having gelatin at home and putting in the fridge, and we become with a solid that has high content in water. But why we chose this specifically? Well, first, of course, because this high content in water it's very similar to our body. It has high porosity, and it's biocompatible with our body. Other characteristics, such as low stiffness, which resemble the stiffness that we have in a bone marrow, are essential, and it's very easy to chemically graft, meaning that we can give nuances to this material to interact with the cells. So, having the alginate as a central point of my thesis, I divided this in three different branches. The first branch was to study, in chapter 3 and 4, the 3D cell biology. So going deeper in the cell biology and molecular biology on how to understanding how this 3D environment would change the cells. The second was the biochemical interactions. In this one, we wanted to have something more complex, and instead of just seeing what the cells behave, actually making change to the mesh and see how the cells will respond to that. And finally, in chapter 6 and 7, we wanted to know something about the cell secretome, meaning the proteins that are, for example, secreted by the cells when they are encapsulated in these systems. So the starting point in chapter 3, we wanted to compare aggregates, cells grown as aggregates, with cells grown in alginate hydrogels. We reached the conclusion that the alginate hydrogels showed a better cell survival, and the cell viability decreased over time for all the systems. But the more interesting fact about these systems and both the alginate and the aggregates, or 3D culture, is that the cell cycle was halted in the G0, G1 phases. But what is that cell cycle? Well, cell cycle is a normal behavior of the cells. The cells are always cycling in our body. They go from G1 phase to S phase, G2 phase, and M phase. When they are not doing this, they become in G0 phase. And this can only happen in 3 different manners. One, they get senescent, meaning that they are dying. And the other, they get differentiated, meaning that they get specialized in some other tissue. There is a third one that is quite special and belongs to stem cells usually, which the cell becomes in G0 and is able to re-enter the G1 and re-enter the cell cycle when needed. This is very important to study what a quiescent adult stem cell is, and we define 4 parameters that we thought they are extremely important. One is that the cell has to have long-term viability, has to be multipotent, like I said, needs to differentiate or specialize in different tissues, and of course non-apoptotic and cannot be proliferating. So that's what we did in chapter 4. We did check that the cells were multipotent, and we saw that by the differentiation towards the 3 lineages of the classical 3 lineages of mesenchymal stem cells. We saw that we have long-term viability by these green dots here that show that the cells were viable throughout 28 days in culture. And in this culture, we can see that there is a lack of red staining, meaning that the cells were not proliferating, were not dividing. Well, taking this into consideration, we went further into it, and we wanted to see why this was happening. Well, for that, we saw 3 different quiescence genes that have been described to be present in other quiescent stem cells, such as muscle stem cells, for example. And we realized that these genes were upregulated in our 3D system. Then we reconfirmed that the cells were indeed in the G0 phase, and they were not apoptotic. But there is another important aspect of quiescence, because recently quiescence has been divided in 2 different states. One that is called quiescent alert, and the other that is called deep quiescence. And this distinction has been made by the expression of the mTORC system. The mTORC is this very complex group of proteins, but I want you to pay attention to Raptor protein. Without Raptor, the mTORC doesn't work properly. And we realized that in our 3D systems, there was a lack of the Raptor protein, meaning that actually our cells are in deep quiescence. Well, moving forward, we wanted now to see behavior, but by changing the mesh of the alginate. We did that by selecting 5 proteins that are available or present in the stem cell niche. And with those proteins, we chose peptides that have the active part of the proteins, and these are proteins related with cell-cell interactions or ECM cell interactions. We attributed a color code to each of these protein or peptides, and with that, we did all the 64 possible combinations, which originated 64 different biomaterials based on an alginate that we exposed the cells to. Well, because this is quite a huge number of different conditions, we came up with a high-throughput screening device house-made that was then analyzed in an automated way in the confocal microscope. With image processing using Cell Profiler and Python, we came up with the results about the cell shape and the cell proliferation. The main results of this chapter was that, for example, condition 32 has round cells and no proliferation because we see lack of red staining, whereas, for example, we have completely different condition 27, where cells are indeed positive for proliferation and have a spindle-shape-like structure. So we plotted these two results, eccentricity, which means how round or as an ellipse the cells are, and proliferation, and we concluded that there is a positive correlation between these two. So the more ellipsoid the cell is, the more positive for proliferation the cell is. Moving forward, in the final branch of my thesis, we wanted to look at the secretome, so the proteins that are secreted. And we started that by a hint that we had in our lab that staniocalcin was being expressed by mesenchymal stem cells. And staniocalcin is a protein that has anti-inflammatory functions, reduces apoptosis, and aids in angiogenesis. So it does this by connecting to other cells, and then does activate those cells and can be seen as working as, for example, a pro-inflammatory protein. We realized that a decrease in focal adhesions, it's something that leads staniocalcin to be secreted more, as you can see by these cases in here. And what are focal adhesions? Focal adhesions are the proteins responsible to connect the cells to the ECM. So when we don't have these focal adhesions, we have secretion of staniocalcin. Interestingly, we have a lot of focal adhesions when cells are grown in a 2D surface. But when they were grown in 3D surface, in a 3D environment, such as alginate, there are not a lot of focal adhesions, which led, again, to increase into the secretion of staniocalcin. So, but this is just one protein. But what happens with much more secretion of other proteins? For that, we intended to mix our hydrogel with electrospun fiber, which would protect the hydrogel environment, and we wanted to see the secreted proteins that were coming from these cells. So first we were able to do, and we baptized this as a hydrocup system, the junction between these two materials. We managed to have different thicknesses, and we saw that these cells inside of this system were alive throughout 20 days of culture. Then by PCA analysis, we realized that, for example, thin and thick hydrocup are very close in their secretion, and they are not so far from the alginate by itself, but these three conditions are very far in terms of identity to the two cells grown in 2D. Finally, the objective of this chapter was indeed to have this implanted in a tissue, and we did verify that the cells are present after 3 weeks in the animal model. This reaches to our conclusions, and the take-home message are that 3D hydrogels did induce HMSC behaviors changes, and this was in proliferation, quiescence, cell shape, and protein secretion. These 3D hydrogels may be very interesting for wound healing purposes and for any application regarding that. And I think that by looking at the native tissue and get inspired to do artificial biomaterials will probably be the future and successful of biomaterials. And also Mario agrees with that. And with that, I would like to acknowledge my supervisors, Lorenzo and Sandra, Cell Library, which was my team, the people involved, the animal experiments and data analysis, Merlin, my parent that are here, the funding agencies, and you all for your attention. Thank you so much, and I give my word back to the prorector.
Thank you for this very interesting presentation. I wish you good luck with your defense now. And the opposition will be started by Professor van Griensven, Professor of Regenerative Medicine in this university.
Thank you very much, Prorector. Dear candidate, first of all, I would like to congratulate you on a fantastic work on the study, and I like the Super Mario, so that is very nice. Also, my congratulations to your supervisory team, of course. I would like to start the discussion with chapter 4 with you. You stated that your hydrogel mimicked the bone marrow environment, and therefore you had this quiescent state. However, when you use alginate hydrogels, they are often used also for chondrogenic differentiation. So, having in mind that you have an alginate hydrogel that have these two properties, what is making the difference of staying in quiescent MSC or going into differentiation of a chondrocyte?
Highly esteemed opponents, thank you so much for your comments and for your question. That's a very interesting question that you are asking. I think that the difference between that is a ratio between the viscoelasticity of the material. For example, it has been shown recently, if you have the ratio between G double prime and G prime to be 0.2, it means, for example, that the MSCs inside it are more prone, for example, to go towards chondrogenesis, and therefore have space to modulate the environment and create space enough to do the pericellular space to be a chondrocyte. Where here these properties were not exactly the same. We didn't reach this 0.2, and it was very strict environment. So we have few RGD anchorage points. Also, RGD, it's important not to be present for chondrogenesis because the cells do need this pericellular space. So we kind of avoided this situation. And then we, of course, showed that was not happening because we saw markers for multipotency and saw that the cells coming out from the hydrogel were still multipotent as well.
That's interesting. And going on in that, when you then look at different stem cells, so you describe here the bone marrow, but you also have stem cells like in adipose tissue and all kinds of other tissues. How do you then see that? Because those environments are also quite different from the bone marrow.
Can you? I didn't understand.
So you have different niches in the body. Yeah, and so the bone marrow is really more this liquidy situation where you say, okay, that's important for your quiescent state of your stem cells in the bone marrow. However, you have also like adipose tissue, which is much more dense tissue, which then goes in another direction in terms how the hydrogel, how you describe it. So do you think that this environment you created is typically only for bone marrow stem cells? Or when you would have other stem cells, does it also count for that?
Well, that's also a very interesting question. And in reality, most of the stem cell niches do have big resemblance. They are very similar in constitution and as in mechanical properties specifically. That's what you are touching in your question. It is known that most of them, for example, the niche in the brain also has 200 Pascal of mechanical properties. It is known there's a lot of common proteins that are present in most of the niches. So in my chapter number 2, that's where I did a revision of the properties. And for me, it was really interesting when I was reading this to realize that there is a lot of common properties, like I showed, between different niches. I'm not sure exactly, and because I think there is not much information about that in the literature, what are the differences between, for example, saying mesenchymal stem cells within a niche of the bone marrow or mesenchymal stem cells in a niche of the adipose tissue. I cannot tell you what those differences are and how that would influence. But I do believe that there are certain key elements to all these niches that are essential to maintain the stem cells to be there and to be in the quiescent state and live throughout our lifespan. And that they are
also different from differentiated states.
Exactly.
Okay, thank you. Then going on in chapter 5, on the concept that you are now also describing, you showed very nicely that the shape of your cells did change, and that when they were like more flat and spreaded and sitting there was more proliferation. I found it intriguing. So which mechanism do you think is involved in that? When they are sitting there and having more contact?
I think that, I wish I would have an answer, like a certain answer to that, but I don't. But I hope one day we will. But my feeling is that there are two things. One, it is already established that surfaces that have more anchoring points for formation of focal adhesions will lead to more proliferation. This is something that has been established in 2D. In 3D, this is something that is difficult to understand precisely because we now do realize that even soft materials, if they have proper rearrangements of the anchoring points for focal adhesions, can promote more proliferation with this elongated shape. So indeed, like in 2D, if you have a very elongated shape, you will get more proliferation. For us, it was striking to see that somehow this combination of different peptides are working towards having this spreadness and then the proliferation. For me, it's quite obvious in a way to see that the more elongated shape, even in 3D, will lead to more proliferation.
And do you think that has something to do with the molecules that are important, like the focal adhesion, like integrins, things like that? Do you think that is important?
Yes.
And do they differ for those cells, for your quiescent cells? Do they have a different mark setup of integrins than other cells?
I think, well, we used several different peptides that interact with different integrins. So, for example, we have integrin alpha-5, beta-1 for our RGD, or we had, for example, the collagen type 4 peptide that we use that interacts with CD44. So it is difficult with it, and that was the precise idea, which was to go beyond trying each and every single peptide and try a mixture of them and see what would happen to the cell. So we cannot identify now exactly which point or what happened or which integrin, but I am very confident that indeed must be something downstream that happens after the integrin interaction with the peptides that are exposed in the alginate, and that will lead to changes in the nuclei and to expression of quiescence genes and proteins.
Sure. And when you then look at your peptides you used, your ECM peptides, think of collagen. So in the human body, when you have an injury, you get exposed to collagen, and that actually elicits a whole mechanism of like repair, regeneration, etc. So when you now expose your stem cells to the collagen and they get more quiescent, I found it intriguing, because I would also think that make the cells more active and want to regenerate actually. How can you explain that in your niche?
Well, that can be explained as well by two different I will give you two different parts of the answer, let's say. One is that actually it's been shown that the fibrillar structure of the collagen, when it gets more loose, it promotes, it gives a signal for cells to be more proliferative, but we are talking about the whole collagen and not just the peptides that we chose here. So the peptides that we chose here, we know for collagen type 1, for example, they interact specifically with a sort of integrins, 2 or 3 integrins specifically. Whereas, for example, collagen that has been broken down during inflammation process, for example, releases cryptic sites of interaction with integrins. For example, RGDs show up when there is this degradation that will indeed promote cells and then probably the cells proliferation as well. In here, again, it's very difficult to pinpoint exactly what is contributing to what. I think that now we would extrapolate for the best conditions and then go in detail and block each one to see what happens.
Thank you very much. I'm very satisfied with the answers, and I give back to the pro-rector.
Thank you.
Thank you very much. The opposition will be continued by Dr. Barrias, a scientific investigator, Universidade do Porto.
Thank you very much. So I address my greetings to the pro-rector, to the other members of the jury, and also to the supervisors, Lorenzo and Sandra, to whom I thank the invitation to be here today. I'm very pleased. Of course, I'd like to congratulate the candidate, David, for reaching this milestone, a very important one, and for being here today. For sure, a very special day for him, his family, and his friends. So, David, thank you very much for the very clear presentation. Congratulations for all the hard work you have developed over the past years. It has been truly multidisciplinary. So you went from polymer chemistry to scaffold development to cell and molecular biology, high throughput, in vivo studies. So this has been a major effort and it deserves high recognition. So thank you and congratulations. That said, I will now jump into a couple of general or more specific questions about your work to get your perspectives on some of the subjects. And I think I will go directly to the mechanical properties, which was an important focus of your studies. And I will start by asking, this is a bit provocative, but I will start by asking you, in your opinion, how can we define a soft and a stiff hydrogel? What is a soft and a stiff hydrogel? I know you have discussed this in your thesis, but I would like to hear your opinion on this, because this is still a bit controversial. And also following this, if you think that stiffness is the only parameter we should look at when characterizing cell response to their mechanical environment.
Esteemed opponent, thank you so much for your comments and kind words that you just told us. So your first question related to defining what are the a soft and a stiff hydrogel is indeed very provocative, because well, I would say there's different opinions about it, and I would say in terms of values in Pascal, let's say, I would say that the soft hydrogel goes from 0.2 kilopascal until 1.2, something like that. After that, until 3 or 4, until 5, actually, you will have a medium stiffness hydrogel, and beyond that, probably you have a harder, stiffer hydrogel. However, and that comes to your second question, I think that is very outdated, honestly, from the results that we have now, to think only and like that to that question, to address that way, because indeed, I don't think it is not the matter of the mechanical properties per se, but the force that the cells are able to exert on the matrix. Meaning that, for example, that you can get one hydrogel that you go to the rheometer and you have 5 kilopascal of properties, but then the mesh is so tightly bounded to each other that it's not elastic to be pulled by the cell. So the cell is not so if the cell doesn't pull it, it doesn't create a focal adhesion. If it doesn't create a focal adhesion, it doesn't feel that is a soft, or it will feel like a soft material and not like a hard material. So it will, at the end, be a mixture of both. I think we need to get beyond that and realize that materials do have to interact in a way that allows the cells to pull like they pull the fibers in collagen. That has been beautiful videos showing that.
Thank you, David. So a bit following on this question again, you suggest with your studies that the mechanical cues can even override the effect of biochemical cues, and then softer matrices induce quiescences. This is kind of a take-home message. But for instance, in other systems like organoid cultures, very soft materials which are more permissive are actually needed for these structures to proliferate, remain highly proliferative, and grow. So my main point is, do you think that we can generalize these conclusions on the effect of mechanical properties on cell behavior, or rather we should consider them to be largely material dependent or cell type dependent, or even cell density dependent? What is your view on this?
I think it has to be native light dependent. So we need to look to what we want to create, the microenvironment that we want to create, it's specifically to which individual one. I think that's how we should design materials. Because again, the example that you gave, if we use a fibronectin hydrogel, we know for a fact that the cells will be able to degrade the material over time, and that will promote them to migrate, because first is degradation, then migration, then proliferation. So, you know, like I think that in that sense, we need to look specifically of the characteristics and know, okay, this material has to last for this time, this has to exert this function, we want this to organize as a body of cells to do an organoid inside of this material, or we want it to colonize the entire material, and then we have cues to be enzymatically digested, or we want cues for the material to exert these mechanical properties now, but more or stiffer over time. I do think that the idea is always to look at the final application of it.
Okay. And in the type of hydrogel system that you specifically use, which are alginate hydrogels, and as you know, they are non-degradable, at least during the time frame of the culture that you used. You will have a physical barrier for cells to spread. So these hydrogels will physically constrain the cells and they will force them to adopt this round shape. Do you think that this could indeed be the main trigger for inducing the quiescent state that you saw, or and not so much the mechanical properties? Could you elaborate a bit on this?
That's a very good question. I thought so, and I was puzzled by it because also we have alginates with more contents of RGD and less content of RGD, and then with less or more alginate on it, and we kind of have the same sort of results. It always leads to more or less cells that are round. It can be different, of course, and again, that depends on, for example, if you mix high molecular weight with low molecular weight materials, that also changes the plasticity of the material. But the thing is that we culture cells in collagen and we got the same results, and collagen should be very permissive in terms of, well, it first will be degraded by the MMPs of the cells and should have more space, especially because that was the one with lowest mechanical properties. It reached the 0.2 kilopascal in the rheometer, and I was using that gel as a positive control for proliferation. I did realize that it was still inducing the cells to be quiescent and with the same quiescence genes. So yeah, that's my answer, I guess, for the question. I'm not sure if it is just the entrapment. I wish it was, because then I'd make my 3 or 4 years around this chapter 1 year, but it was not.
I agree with you. It's a far more complex effect. So I'll probably now jump into the hydrocup just for a very brief question. So I totally understand the aim of the tool, where you could actually maintain the hydrogel with cells in specific tissues where mechanical stresses could be more important or more relevant. But for me, one big concern was the fact that you are adding a very big diffusional barrier around this hydrogel with cells. So adding a diffusional barrier that will probably make it very complex for the nutrients and cells to go in, and vascularization to happen, and the cytokines to come diffuse out of the construct into the tissues. So I know you discussed that you can change a bit the properties of this cup, but what are your big strategy to?
Yeah, indeed, this is, as a disclaimer, was a prototype experience, of course, of an attempt to do something like you said, as a protective shell for the hydrogel, because, like you said, the hydrogel is not degradable by our body, but it is, the calcium within it is replaced by phospho ions over time. So we have degradation of the hydrogel. So that was the idea, is to protect it even more and the cells to be in that 3D environment inside. But I think that we could play with the thickness, indeed, of this membrane, even though we saw that some cells would get out of the system and would be populating the outside. So I think that this would aim for something that is short term, so like weeks or 2 weeks, for example, for cardiovascular issues, like an infarct or something. And in that term, we could, for example, functionalize the outer shell of the hydrocup with some sort of materials or molecules that will, for example, either prevent the entering of other molecules, or allowing the molecules that are inside to get out more easily. But we did show that they are still able to secrete. One thing that we wanted to know and this PCA analysis showed us that indeed there is is a difference even in the secretion between alginate and the hydrocup itself. And now I think we should go more into details and see what are the proteins that are getting out or not getting out. Are these proteins relevant? Are these going to contribute for the homeostasis or to the regeneration of whatever tissue we aim to? So, to answer your question, indeed, it was something it was an idea that sparked on getting them together and see how they would interact. And these are very tiny membranes that we performed in the lab. The application was that. I think that the whole brainstorming around it still has to happen.
Okay, David, thank you very much. I think I run out of time and I'm very happy with your answers. So congratulations again and thank you.
Thank you. The opposition will be continued by Professor van Griensven, who will ask the points from Professor Fibbe, who could not be present here. Yeah.
Professor Fibbe also stated that he is very happy with the booklet and congratulate you and the supervisory team. The first question he had was a more general one. The term mesenchymal stem cells is used to refer to stem cells and sometimes also to more differentiated cells, and therefore the term mesenchymal stromal cell is sometimes preferred. Then you also have the term adult stem cells, and the word adult is used to indicate the origin from adult tissues as opposed to embryonic tissue. So, a true stem cell combines the ability of self-renewal and multilineage differentiation through an asymmetrical cell division resulting in one stem cell and one more differentiated cell to mature progeny. So when you have this ultimate criterion to self-renewal, you have the secondary repopulation ability in vivo. So do you think that your mesenchymal stem cell meet this criterion, and what is the underlying evidence?
Highly esteemed opponents, thank you so much for your question. And for the compliments, of course. What Professor Wim described, it's indeed all of that for stem cells. And it's very controversial about mesenchymal stem cells / stromal stem, mesenchymal stromal cells. It's very difficult for me to say this. But the last criterion, indeed, and that is something that has not been shown for mesenchymal stem cells, is when you implant them into an animal, you have indeed the giving rise to a progenitor inside of the animal, but you also have another stem cell coming from that with markers, so you know that it came from that one. And then you are able to re-implant that in the other animal and give rise again to the three classical lineages of mesenchymal stem cells. This has not been performed in human stem cells. It has been performed in a specific population of rat stem cells. I'm not sure if it was rat or mice stem cells. And this was a specific population that has been sorted according to specific markers when they realized that nestin-positive stem cells were the ones that could actually do such a thing. Our stem cells became positive for nestin, became positive for the nestin marker and for STRO1 marker. We did not try this, so I cannot answer if it would happen or not. I would love to see that happen, but it's indeed something that should be done very quickly so we could maybe move forward to the definition of what the stem cell is.
So when we follow on this idea, so your quiescent stem cell in your 3D culture, you say there's deep quiescence, right? Are they really able to differentiate and have the quiescent population and at the same time? Or do you think it's more a quiescent progenitor cell that lose the quiescence when they start to differentiate? So the cells are in there, are they able to have this?
Well, there are two things to that. One is that I believe that there are some protocols to induce quiescence, specifically the starvation that we did, and the 3D hydrogel system that sorts cells out. This because the mesenchymal stem cells that we have are a harvest pool of stem cells that came from bone marrow, meaning that we have all sorts of different stem cells, and there is no specific marker, like for example in hematopoietic, you know that CD34 and CD38 will define as one specific population of quiescent and deeper quiescent stem cells. So not having that, I think that these culture systems will sort the cells, and I guess that some cells inside of those systems will maybe be the ones responsible to give rise to the deep quiescent stem cells. Of course, and what I cannot say for sure if it's cells that are already in the progenitor level that will give rise as well. They will survive and will give rise as well to the differentiated cells, or even if, because we know that the cells that differentiated, not all of them differentiate when we do the protocols in 2D. So maybe some of them are indeed the quiescent, in deep quiescence, and we don't know because we didn't check for that.
And then one last question. So imagine that are really stem cells.
Yeah.
So they go into one stem cell, and then another one is for the differentiation. Do you think in such a 3D culture that you have, is that possible, especially when you think of different requirements like oxygen tension for the quiescent cells, and on the other hand, other requirements for the differentiating cell?
So if I believe that they have those conditions.
Yes.
In my hydrogel, I believe they don't have those conditions.
Mm-hmm.
Well, no, actually, let me rephrase it. I do believe that in my hydrogel, they have those conditions because we have seen that the cells that are able to get out of the hydrogel have populated the surface of the hydrogel and form sometimes at the end stage a thick layer of cells that are spindle shaped like, and they are proliferating. So when we saw that the periphery of the hydrogel is the one that of course has more oxygen, more nutrients available, and perhaps that's already a trigger for them to become proliferative, and the ones in the core, such as happen for example for aggregates, are the ones that become quiescent. So I think so.
I think Professor Fibbe would be very happy with the answers. Thank you so much. I give back to the Pro-Rector.
Thank you. The opposition will be continued by Professor Welting, who is Professor of Molecular Cartilage Biology at this university.
Thank you. Dear candidate, congratulations with a beautiful thesis. Of course, I also read it with a lot of enthusiasm, and also congratulations to the rest of the team. We are not only here to say brave and nice words, but also to see, of course, what you think about all the questions that we have for you. So I would like to go to chapter 5 first. Mm-hmm. So, and actually this is more like a technical question maybe first. So if you are setting up such a high throughput, screening system, there are a lot of things, of course, that you need to take into account about, the reproducibility, the quality of the peptides that you incorporate. Are they still active? Are they still intact? These kinds of things. And I was reading through your thesis, and I did notice, of course, that you drew those NMR spectra where you are actually seeing the phenyl or phenylalanine group, I think, as an indicator of the presence of the peptide. Yeah. But I was not yet so convinced about, whether you did everything actually to ensure the quality of the peptides that are incorporated, or even the quantity. So could you allude a little bit more on what you did in order to make sure that you knew actually what you were working with?
Okay. Highly esteemed opponent, thank you so much for your comments and for your question. I think that we didn't do all that we should have done in determine the presence of the peptide. I think more analysis now should be performed, but we did know that there is for sure an effect that comes from the presence of for each individual peptide in there on the cells. which for me it's one of the most important evaluations to see, because we saw that, for example, we have alginate that was that we attached norbornene to it, and to that we did click chemistry with the peptides with the cysteine group, cysteine amino acid. And what we saw is that there is an increase of the phenylalanine peak, like you said, in the NMR, but there is also a decrease of the peak the peaks that show for the norbornene. So we know that a reaction occurred between these two entities. We just make a quantification of the amount of peptide that is actually present, and we made sure that this peptide is really attached and not just in the solution when we did the NMR analysis. We should have done more analysis to prove it, but we also did, for example, each individual peptide with the alginate in 3D was cultured with cells, and we saw an effect. Compare that to alginate with norbornene, and we saw that norbornene didn't do anything to the cells, and each individual peptide has made a change in the shape of the cells. So, but in that, we should have seen that the focal adhesions have been formed. We should have blocked each of the peptides' communication to the cells to prove that this interaction was driven by the peptides themselves.
Right. So what would be your most important? So if you could work on this again, so what would be the most important quality aspects that you would like to check then?
Yeah, first of all, I would repeat all the chemistry reactions, because this click chemistry could give rise to different yields of the reaction, and that for me will be very important to have that standardized. The other thing that I would make sure is this one that I said of the blocking. So I would block all the receptors for the peptides that we have included and see if that will make changes in the cells.
Find a causal relationship, you mean?
Sorry?
To find a causal relationship.
Yeah, that's what I would do. I think for me those are the two essential biological assessments that I will do to the.
Okay, thank you. So moving on to chapter 6, and I will keep this short in order to give the other opponents also the chance to interrogate you. So chapter 6 is about staniocalcin. Yeah. And so an interesting molecule, and you actually show that it is somehow responsive, to the mechanobiological aspect that you then apply on these cells. Yeah. There's YAPTAZ involvement, so that's fine until then. But I was a bit puzzled in the end, as in, you show that the expression of the protein is being regulated in your cells, but I was left a bit in doubt as in what the protein is then doing, because if I read the introduction, it seems to be doing like almost everything.
The staniocalcin.
Well, yeah. So it has many functions.
Yes.
So what do you think staniocalcin? Well, why do you actually think it is really interesting to study staniocalcin in the context of your cells? And then maybe like the sub question would be, why is this important for your tissue engineering approach?
Yeah, so indeed, the staniocalcin has been involved in several different mechanisms, like you said, and specifically also seen as a major target now for cancer, because it's always promoting the survival of the cells and the proliferation even of the cells in cancer cells. But we getting out of that gigantic field and going to tissue engineering. That's what's our focus. We saw that staniocalcin was interesting because it showed in a lot of secretomes from other publications. And when people see and make the analysis of the secretome for a material that they are producing, and they say that there is pro-regeneration effect on whatever tissue or system that they are doing, the staniocalcin appears
that's
very
what I read. Yeah.
Constantly. It's always showing up.
But what do you think it is doing there?
I think it's pro-angiogenic. It's proven that it's pro-angiogenic. I think it works as an initial regulator for showing that something is wrong with the tissue. So to signaling that there is a wound and this needs to be fixed, especially because of other stem cells. And I say this because since it has been shown by Prokop that the staniocalcin is highly connected to vinculin in the focal adhesion. So when focal adhesions disappear, which is connected also with the wound, this is released to the environment, and I believe that it might be the result that mechanism might end up making the regeneration.
Okay, so I learned that I need to learn a lot more about stenocalcin. Thank you.
Yeah.
So I give the word back to you.
Okay, thank you. The opposition will be continued by Dr. Tamar Sebi, principal investigator at Merlin, this university. Yeah.
Thank you very much. Dear candidate, I would also like to join others to congratulate you on finalizing your research and delivering your thesis, and also to extend that congratulations to your promoter and co-promoter, of course. I, of course, have some questions for you. I enjoyed very much reading multiple chapters of your thesis, but I'm going to focus right now on chapter 5. And to start with, I would like to challenge you on your study design. You presented that in your presentation earlier, and it's also on page 137 as the first figure of the chapter. What you have chosen is to modify your alginate with different peptides, and then you have made combination of these peptides, and you have gone for a throughput approach where you actually nicely put all of these different materials in one platform and analyze that. Very nice. My question is that why didn't you take a different approach in which you would first look into the effect of individual modifications, and then based on that design an optimal material for controlling the proliferative state of the cells? This would have made your experiments smaller, but you have chosen to go for a more elaborate one. Did you expect some cross interactions between different peptides that you could not have seen them in the individual assessment of the peptides?
Esteemed opponent, thank you so much for your kind words and for your question. I think we just went bold on that approach. Let's be honest about it. I think that the entire goal of that chapter was more related with making a mixture of something that has not been happening. Of course, that what you described it's how it should have been done, or let's say it's how every study of peptides is conducted. And it's something that I would like to do it now for specific conditions. So we will rather have something that is more within randomness of picking these peptides, mixing them together, and see one condition that really works, such as all other aspects of science. For example, the topo chip that you are familiar with. It's also random structure, and one structure because we don't know why, but we will know it afterwards. So it's basically the strategy is to start with, okay, we have one condition, and then now let's understand why this condition is nice. And that I think that was the strategy of that chapter, and not the other way around.
Okay, that's acceptable. That's a strategy that I also like very much. You might indeed come across some effects that you did not anticipate if you made the study smaller. Let me just bring one example of that I just came across while I was reading the chapter now. So you have chosen two conditions that they had a low EDU expression and one that is higher one. And then you have identified these on different or two sides of the spectrum of cell morphology, correct?
Yeah.
So conditions 32 and 33 are basically low EDU, and condition 27, you know it by heart, is at the high EDU side of the spectrum. Okay. If I look at, for example, 32 and 27, one of the differences between them, so they have some similarity, so they have both of them one RGD in there, and then they have the peptide called HAVDIG. And then the difference is that 33 has an additional peptide. Yeah. Then based on this, logically, I would conclude that probably that one peptide is the reason why you have less EDU-positive cells. So I hope I could actually follow your logic correctly. So yeah, please correct me if I'm wrong. So if I look at only that one peptide in your study design, that's peptide number 3. So condition number 3. And if I look at condition number 3, it falls really at the beginning of low EDU. Right, so it does something to the cells that, yeah, it reduces.
Or it doesn't do anything to the cells because it has low.
Exactly, it's similar to a condition that you have actually no modification.
No peptide, yes.
Okay, And then when I look at cell morphology, it falls somewhere in between.
Yeah.
So it is not exactly the morphology that you actually identify for the other two modifications.
Yeah.
How do you explain that?
Well, I will explain that as a soup, and you put a lot of different vegetables in it, and it gets a final flavor to it, you know. And this is a funny way of explaining, but I don't know. That's the answer. So why you put several different vegetables and then you get that flavor? And maybe that's the same here. I will have to go to look at. What I will do now, it's going to probably the 5. In each, in the bottom, at the top of that scale, and in the middle, and see what are the similarities between these. Why these peptides are doing this? I'm going to block each of these peptides. I really don't know how to answer because I was also puzzled myself when I look at it too. Those are the conditions that are statistically different. That's why the 27, 32, and 33 are the ones that I pointed out and are very different. The rest of it, I'm hoping that now with a deeper analysis where we have more things in consideration, because here you can see that there is the length of the diameter, but we studied several other aspects of morphology that we will include now for the next round of analysis, and we will correlate that with the EDU as well. So it might be that other aspect of shape or a specific peptide that interacts with another peptide and gives a complete different reaction to the cells occur. So I hope I will tell you soon, like in the next year, maybe.
I really hope you do that. Okay, so maybe just one additional question on this chapter, just to challenge you. What would you do next if you had more time in your PhD to actually increase the validity of these results and improve the setup? So what did you identify after you saw all of the results that should have been changed from the beginning?
Yeah, well, first of all, I will increase the high throughputability of the system, as in the device, it's all interesting and all, but I think it should be an automated way of putting the gels inside. So we fixed our high throughput analysis in the screening and in the obtaining the data from the confocal. That was really interesting to actually get, but I think that now I will focus more on obtaining nice hydrogels that are formed properly, always with the same amount of peptide, and then those hydrogels I would focus on mixing them with the cells in a machine that will be in an automated way. Also because to not making it only just for these applications, but other applications. There's another thing that I would like to do, which is, since this is a photo-initiated system, it is possible to mix the alginate with a cocktail with peptides that we want and with the cells, and this is it's called it's biocompatible click chemistry. So maybe I will also do that approach on putting the peptides put all together, and then you just put a droplet of each peptide on the gel that you want. You put the light on, and you should have the cross-linking directly to the Nord-Borning entities that we have there. I think those are what I would do. If I had more time, I would do that.
Okay, thank you. I think in light of time, I have to actually give back the word, and yeah, I'm satisfied with the answer.
Thank you so much.
Thank you. The opposition will be continued by Professor Gieselbrecht, Associate Professor at Tissue Morphogenesis at this university.
Thank you, Mr. Prorector. Congratulations, first of all, also to your supervisor team, and on this very nice achievement. I enjoyed a lot reading your thesis. However, I still have a few questions that I would like to discuss with you. But first of all, I would like to get some more background information from you about kind of maybe the developmental origin of mesenchymal stem cells, whether you can give us some more information about that. Is there something we can actually learn from that? Because, well, we all know MSCs are quite a heterogeneous population. There are a lot of kind of problems associated with that. We heard about kind of donor variations, for example. Can you maybe go a bit?
Okay. Highly esteemed opponent, thank you for your questions and for your comments on my thesis. We had two different donors actually used during this thesis, which is also something that maybe has passed by without notice. But chapter 3 has been conducted with one donor and the rest of the chapters with another donor. But this is an important fact to know that we got quiescence in both donors in different studies. Taking that out of the picture, because I think that's a very important comment to do, the origin was always bone marrow for both donors, coming from different companies and different places of the world. And it indeed is a very heterogeneous population. The selection of this population has been done by adherence to plastic tissue culture plate, which again, I don't defend it, as you see in my thesis. I think it should have, another thing that I would love to do is to get population that is already sorted through a specific number of markers. And then these cells were identified by the CD45 positive cells, positive for STRO1 as well. These were the essential let me just, I'm saying something wrong. I think CD45 negative, I'm a bit confused. It's CD90 positive, CD45 negative, and STRO1 positive. And these cells were, so then they were checked for the differentiation. But indeed, it's still a pool of different stem cells. I don't know if I answer your question.
Yeah, maybe going back to the developmental origin of of the MSCs. Are there any other sources that you could use actually instead of using cells that you have
If I may just answer, or thank you. There are several different options and different sources for stem cells. We could have get stem cells from the umbilical cord, for example, from adipose tissue, and those cells could indeed behave completely different. So one of the things that in the future this should be checked and should be should be more consensual is to see if all markers correspond always to the same stem cells, regardless of the niche that we extract the cells from.
Okay, thank you. You can go on forever, I think.
Yes.
But the time appointed for your defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense, and I request that you and your company await the results of our deliberations and our return in this room.
Thank you so much.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Southside, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
David Boaventura Gomes, the degree committee here present has assessed the quality of your thesis and your defense. And in view of the positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Moroni is authorized to confer upon you this academic distinction in accordance with Dutch university custom. And I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity, at all times to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law, and in conformity with the decision of the committee here present, I hereby confer upon you, David Boaventura Gomes, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and all the other members of the committee, and affixed with the official seal of the university. Dear Dr. Gomes, dear David, as always in life, patience is of key importance. With a lot of patience, effort, perseverance, and resiliency, you have finally come to the end of this doctorate journey. It has been a special pleasure for me to see the man that you have become, a brilliant thinker, a creative mind, combined with the right pinch of a critical mindset that actually is what makes training into a scientist and what requires in general for professional development. You and I go back in time quite a while. It was more than 10 years ago that we met in a nice, small conference in Leiria. At that time, you were just finishing your master's degree, if I remember correctly, after a good experience in the lab of Fabien Guillemot in Bordeaux, who happens to be one of the most successful founder of a biofabrication company nowadays called Poietis. So also quite a nurturing experience. Supervised by Cristina Barrias, with us today as well online. You then embark into a career also in the animal facility of the University of Porto. Yet we never lost contact, actually. At that time, in 20, I had no PhD position available, but we both kept talking about science by email, almost a sort of epistolar relationship of other times, which reflects also one of your features, combining all traditions and values, taking time to valorize relationships, something that in this today speedy world has been unfortunately forgotten oftentimes, and which I encourage you to hold on to. I remember nowadays vaguely the content of those emails, but the memory of a pleasant person with a strong passion for science, a genuine curiosity to discover new knowledge. Then an opportunity came. We moved to Maastricht, a new grant came at the same time, and I didn't even have to make a selection. I knew you were the right candidate. You came to see the new environment. You had a lot of courage to take that position, because at that time, about spring 2015, there was nothing. No labs, no office, nothing. We thought it would be only for a couple of months. We were wrong. Nonetheless, it was clear how crucial you were in those days, where labs could not be used, with ethical permissions, preparations for the labs to start, and all whatsoever small, minute tasks that contributed beyond your own, that you contributed beyond your own PhD, to the PhD of your colleagues, and many of the other scientists of Merlin. David never saved up a drop of sweat, would meticulously design and perform experiments tied to bold questions. Mesenchyma stromal cells being quiescent is still today provocative concept that we could not univocally answer with your work, but that surely brings the community closer and closer to unveiling these peculiar cell biological activities. In this experimental work, it was also impressive for me to realize that I never really had a PhD student-supervisor relationship with David. He started with an innate capacity to plan and implement his thoughts, discuss the results and next steps, that it was soon almost as if we were just peers. It is with no surprise that David then became also a precious collaborator for other PhD students and postdocs. Let me also take the opportunity to thank the member of the assessment committee, as well as of the defense committee. They are not the same, and in particular, therefore, I'd like to also thank Dr. Gieselbrecht that joined the defense committee, as well as Professor Van Griesven and Dr. Tamasemi, as well as Professor Velding and Professor Fibe, who was not here today, for having taken the time through your thesis, and apart from Professor Fibe for being here with us today. In particular, I also would like to thank Professor Barias from outside Maastricht University. Christina, thanks for having trained David so well, for your continuous collaborations in these years, which I hope will continue for a long time. Sandra, you go.
Dear David, I could not be happier to see you here today defending your thesis. It has been a very long run with up and downs, intense at times, but also full of fun and memorable moments that I treasure as part of the greatest memories from my time there in Merlin. I joined Merlin in 2017 when you were already a PhD candidate well advanced in your thesis. You welcomed me to the team and helped me get settled at work and also in town, which I highly appreciated. You introduced me to the cell culture lab when I was still working in Croces. I don't know if you remember those times. And you also took me out for more than one memorable night. You have always been ready to open your arms to newcomers and to help making their way easier at the Institute and in Maastricht. With the time, I took on the role of being your co-promoter, and I'm afraid you suffered also the consequences of being my first. During this time, we shared many meetings, brainstorming, intense cell culture hours, and of course, also the wonderful in vivos. You always kept a smile in your face and tried to make the best out of it, cheering us up with your solos and also with your duetos with Jeep, who I believe is also on the audience. I have to admit that I enjoyed every single meeting that we had together. Your capability to brainstorm, to raise up extremely interesting questions, to design experiments, and to look for answers, always in very, very creative manners, was delightful. I saw you growing into a very mature, independent, and highly creative researcher, and I would like to take the chance also to congratulate you for that today.
We would like finally to extend our congratulations to your family, your partner Pedro, and all your friends from Portugal who kept you warm in all these years, worked with you patiently in the last bit of this journey that was challenging combined with work, and listen to you in those discomforting moments that we all have had while finishing the PhD degree. David is a polite, quiet, friendly, yet firm and strong-willed man. David has shown in all these years always very good education, for which our deepest gratitude goes to his parents. It is not easy to find so many good manners and values in a young man. David has been a great example in this respect. Dear David, we wish you all the best for the coming years. Remember the lessons learned in these years in the Netherlands and in the special multicultural and international environment that is Merlin. We wish you to nurture more and more your leading capacity, as you showed us several times in these past years that when you put yourself into something, you start and finishing it with outstanding results.
Thank you so much.
Dear Dr. Boaventura Gomes, also on behalf of Maastricht University, I wish to congratulate you with your degree that you have acquired. I enjoyed your presentation because I have a history in culturing in time of Super Mario 2, maybe Super Mario 1, myelodysplastic bone marrow cells. And I really realized that a lot of things have changed and have improved. I wish you a lot of success in the near future. I wish to give, when finalizing this meeting, I wish to give Dr. Barrias a short opportunity to congratulate also, and then we make pictures and we go to the rafter to congratulate the young doctor. Dr. Barrias.
Thank you so much. I have to say this is also a very special day for me. I know David, as Lorenzo said, for many years, and he's a very special person with whom I also love to work back in some years ago. So I'm also emotional, so I have to be careful with what I say. But David, I'm really happy for you. I wish you all the best for your personal and professional life. You will be always welcome in our lab, and parabéns.
And with these nice words, I close this academic ceremony.
Thank you.